In neurons, actin dynamics are crucial for the elongation, guidance, and branching of axons, as well as the assembly and remodeling of synapses. Axonal branching is initiated by the polymerization of actin filaments to form filopodia that may extend and mature into branches with the help of microtubules. As Michael Sendtner from University Hospital Würzburg explains, this process is particularly important in motoneurons.
These neurons have a huge degree of axonal arborization. So one motoneuron can have up to 10,000 or even more axonal terminals. So there's a very high degree of axonal branching. And when we looked on actin dynamics in axons of motoneurons, we found that the dynamics in the growth cones is completely different from that of branches. Whenever a small filopodium went out from an axon to become a branch, it never survived until there was a kind of very stable actin filament, and this caught our attention, and this was the start point for this project.
Most studies on actin dynamics focus on beta-actin, but Sendtner and colleagues, led by graduate student Moradi, found that motoneurons also express alpha and gamma-actin, which differ from beta-actin by only a few amino acids in the amino terminus. The mRNAs encoding all three isoforms were translocated into axons, where they were locally translated, such that alpha-actin accumulated at axonal branch points, beta-actin assembled into dynamic actin filaments in the growth cone and growth cone filopodia, and gamma-actin polymerized into filaments in both the soma and axon. This is different from the situation in cortical neurons, which have previously been shown to only translocate beta-actin mRNA into axons. Sendtner and colleagues therefore knocked down each of the actin isoforms in motoneurons to see if they had specific functions in axonal development.
When we knock down alpha, beta, or gamma, we found that any of these components had effects on axon length. So beta-actin made a very robust reduction in axon length, but also alpha-actin and gamma-actin had some effects here in this. But when we looked at the morphology, the alpha knockdown motoneurons had a highly significant reduction in branches. In contrast to beta-actin, where the branches were not really massively reduced, but in the beta-actin it was the axonal growth cone, which appeared much smaller and appeared changed in shape.
Using live cell imaging, Moradi et al. Found that the dynamics of axonal filopodia, which can give rise to axonal branches, were reduced in the absence of alpha or gamma actin, but were unaffected by the depletion of beta-actin. In contrast, growth cone filopodia, which support axonal guidance, were still highly dynamic in the absence of alpha or gamma actin, but their movements were reduced when beta-actin was depleted.
From this, it was very clear that alpha and beta and also gamma-actin have completely different function in axons. Alpha-actin is very important at the branch points in order to generate branches, whereas beta-actin is extremely important for very rapid dynamics of the growth cone in order to find its way to the final destiny in the periphery, in the muscle.
Despite this, mice whose motoneurons lack beta-actin maintain normal motor function, suggesting that alpha and gamma-actin can somehow compensate for beta-actin's absence.
When we knocked down beta-actin, there was a compensatory upregulation of alpha and of gamma-actin which was taking place. And the levels of filamentous actin were not really reduced. However, because alpha and gamma had different physicochemical properties, they made much more stable filaments, and they are less fast in generating new actin filaments or breaking them down.
Sendtner and colleagues think that this ability of alpha-actin to stabilize actin filaments is crucial to its role in promoting axonal branching.
Alpha-actin makes these F-actin filaments much more stable. And this actually allows the filopodia really to persist so that microtubules can invade and that this becomes a stable side branch. And we believe that motoneurons need this kind of mechanism in order to generate so many side branches, in contrast to cortical neurons who have only very few, if any, side branches in their axons.
Mutations in the protein SMN cause spinal muscular atrophy, and these mutations are known to disrupt the transport of beta-actin mRNA into motoneuron axons. Moradi et al. Found that the axonal targeting of alpha and gamma-actin mRNAs was also disrupted in SMN-deficient motoneurons, suggesting that defects in the localization and function of all three actin isoforms might contribute to the pathology of spinal muscular atrophy. Sendtner and colleagues are also interested in the role of alpha-actin in motoneuron regeneration, since alpha-actin mRNA is upregulated in response to spinal cord injury. For now, though, you can learn more about the differential roles of alpha, beta, and gamma-actin in axon growth and collateral branch formation in motoneurons in the paper by Moradi et al., published in the March 6, 2017 issue of the Journal of Cell Biology.